Hello everyone and welcome to Instant Biology by Dr. Neelab. The topic that we would deal today is PCR. This is a very important technique and it is used for amplification of the DNA, and in most of the molecular biology labs throughout the world this PCR machine is run on a daily basis for research purposes. So like all the lectures, I have divided this lecture also into a number of parts, and with the help of these questions, with the help of these points, we will construct our lecture in such a way that it would be easy for you to understand. Okay, so let us start with PCR. What is PCR? First of all, let us understand what is PCR. So PCR means polymerase chain reaction. Okay, so now let us understand each word in this full form of PCR. So what is the first word? The first word is polymerase. So people from biology background would know that polymerases are enzymes that are utilized for the replication or for transcription purposes. Polymerase, when you talk about polymerase in sense of PCR, DNA polymerases are used because we want to amplify DNA. We have a template of DNA and we want to amplify it. Therefore, here DNA polymerases will be used. We would talk about DNA polymerases that are used and the components of the polymerase chain reaction in just a minute, in just a few minutes. Now let us talk about the second word. This is chain. So what does chain mean here? Chain means continuous series of reactions without stopping. Okay, one would lead to another, would lead to another. So it is a continuous process that is being conducted. So this is what is meant by chain. And here reaction. This reaction that is being carried out, this polymerase chain reaction that is being carried out, it is not carried out in a particular body, in a particular organism. It is conducted or it is carried out in a tube. It can be an Eppendorf, it can be an Eppendorf tube, it can be a PCR tube. So basically this is not carried out in a particular organism, this is carried outside in a tube environment. So this is basically a in vitro mechanism. Okay, so this is an in vitro mechanism, not an in vivo mechanism, because it is not carried out inside an organism. So this would be clear up till now, I guess. So we have talked about polymerase chain reaction. We have dealt with each and every word of PCR, that is polymerase chain reaction. Now let us go to the need of PCR. What is the need of PCR? Okay, so for understanding the need, just assume a crime scene. Okay, suppose a criminal has murdered a person. Okay, he has murdered the person and the police has come there and they have found out that on the crime scene some hairs have been found or some portion of a cloth has been found, and the police suspect that this portion of hairs or this portion of cloth is from the criminal's body or from criminal's belongings, right? So what do we do? What does the forensic team or what does the scientist team that visit the crime scene do? They obtain whatever evidence that they have got from the crime scene, and then what do? They try and extract DNA from that particular evidence that they have obtained. Suppose in our case, the evidence that the forensic team has obtained is hairs. So, but naturally the hairs would be in a very small quantity, right? So every time going back to the evidence and extracting DNA from the evidence would be a tedious job, and it also would not be permittable. Okay, so once the evidence is obtained, what do scientists do? They try and extract the DNA from it and ensure that the DNA that we have obtained is amplified to such an extent, such a large extent, that it can be used for further checking, for further processes. Okay, so this is basically the need. And suppose you have got, let us take another example. Suppose an organism or a plant is endangered. What do you want? You want to have that the DNA of that particular plant or that particular organism saved. So what do you do? What can you do? You can go for cloning also, and you can again go for PCR also, so that your particular set of genes or your particular set of DNA is amplified, and then you can go on for storage of that particular DNA or particular genes, right? So this is basically the concept, or this is particular reason that that basically ensures, or that is the reason that why we need PCR. What is the need of PCR, right? So again, it is emphasizes upon the amplification. Amplification of the DNA, right? So I guess this would have been clear. Now coming to importance. What is the importance of this particular technique? Okay, so let me tell you that this technique was so important, created so much impact on the scientific community, that its discoverer, who was Carey Mullis, got the Nobel Prize in 1985. So this technique was so much impactful that Carey Mullis, the discoverer of PCR, got, or the inventor of PCR, got a Nobel Prize in 1985 for polymerase chain reaction. Okay, so this was an overview. This was a basic introduction about what is polymerase chain reaction. Now let us go into the details. Coming to components of PCR. What are the components of PCR? So the first component that we have to study is machine. So on your screens also you would be able to see the machine that is used for polymerase chain reaction. In the machine there are slots in which you place your PCR tubes, in which you have filled in your reaction mixture, and then you place your tubes inside the PCR machine and then close it. You type or you input a program for a particular reaction, and then start or run the machine. Then what will happen? After some time your DNA would be amplified. So this on your screens also you would have seen what is a PCR machine. Now PCR machines are also called as thermal cyclers. It is important to understand that why is a PCR machine called as a thermal cycler. Let us understand this. In a PCR machine or in a PCR program, what do we do? There are 3 steps basically, and each step requires a particular temperature for the process to be conducted successfully. Okay, so there are 3 processes. We would go into the detail of those processes in a bit, but there are 3 processes and each process requires different temperature. So PCR machine ensures that a particular temperature, say particular temperature that is 94 degrees centigrade, 55 degrees centigrade, this is the first step. 94 degrees centigrade is required for the first step, 55 degrees centigrade is required for the second step, and then 72 degrees centigrade is required for the third step. So there is a range of temperatures. So the PCR machine ensures that all these reactions or all these temperatures are reached and they are stabilized at these temperatures for some time, and then again for the next process the next temperature is reached and stabilized for a particular duration of time. That is why this machine or PCR machine is called as thermal cycler. Okay, so a cycle goes on continuously from 94 to 55 to 72, again 94. So a cycle goes on continuously for the changing of temperatures. That is why these machines, PCR machines, are called as thermal cyclers. Okay, so we have understood about the PCR machine. Now let us talk about polymerase. Okay, talking about polymerase. When we talk about polymerases, in case of PCR, DNA polymerases would be used. Why DNA polymerases would be used? We have said that the PCR machine amplifies the DNA. That means that we just have to replicate the DNA. We have to make multiple copies of DNA. From one copy of DNA, we have to make millions of copies of DNA. So in this process, what are we doing? We are trying to multiply the DNA content. Okay. So in this process, what do we need? We need a DNA polymerase which would be dependent upon a DNA template, and it would make a copy of the DNA template again. Okay. So this requires a DNA polymerase. But what do you think? The DNA polymerases that are present inside our body, that is DNA polymerase 1 or other polymerases, can be used in this process? No. DNA polymerase of our body cannot be used in this process because they are thermolabile. We require a temperature ranging up to 94 degrees centigrade. Our DNA polymerase is suited. Our body temperatures is approximately 37-38 degrees centigrade, but the polymerase that is required here should be stable at 94 degrees centigrade. So definitely our polymerases cannot be used here. So which polymerases can be used here? There are some bacteria that have thermostable DNA polymerases. The first one is Thermophilus aquaticus. So this is the first bacteria, and the polymerase that we obtain from this bacteria is Tag polymerase. The name of this polymerase is Tag polymerase. Okay. So this is one example. Next one is another polymerase that we can get is PFU polymerase. And again, the third one is Wnt polymerase. So these three are different polymerases that are used in case of PCR reactions. Mostly Tag polymerase is used. These two are obtained from different bacteria. The difference between the Tag polymerase and PFU polymerase also we would take up, but in a bit. So in general, we use Tag polymerase, right? So what are the salient features of Tag polymerase? Let us also understand this. What are the salient features? Features of Tag polymerase. Okay, so let us consider the features of Tag polymerase. Like the DNA polymerases, Tag polymerases also has a polymerase activity. It would polymerize in the direction of 5' to 3'. So the direction of polymerase activity would be from 5' to 3'. No endonuclease activity would be observed. No endonuclease activity. What is endonuclease activity used for? It is used for proofreading, and it occurs in 3' to 5' direction. But in case of Tag polymerase, no endonuclease activity is seen, only polymerase activity is seen, no endonuclease activity is seen. So this is another very important feature. Now let us understand about the stability of Tag polymerase. So researchers have shown that for around 40 minutes, this Tag polymerase can remain stable at 90 to 95 degrees centigrade. At around 90 to 95 degrees centigrade, this Tag polymerase can remain stable for 40 minutes. But as you increase the temperature to 100 degrees centigrade, the stability will drop down to less than would drop down to less than 5 minutes. Okay, so 90 to 95 degrees centigrade would be for 40 minutes, and when you increase the temperature to 100 degrees centigrade, this stability would only last for less than 5 minutes. So you can say that 1 to 5 minutes, right? So this basically is about the tag polymerase, one thing more I would like to add up here, that it does not have a good fidelity. What does fidelity mean? It cannot be used for long polymerization activity, that it cannot be used for a long stretch of DNA. You cannot use for amplification of a long stretch of DNA. It can be used for small stretches of DNA to polymerize them or to amplify them, right? So fidelity is less fidelity. So this is about tag polymerase, basic features about tag polymerase. And when you talk about PFU polymerase, we have already talked about PFU polymerase. It has a higher fidelity when compared to tag polymerase, right? So this was all about the polymerase. Now let us come to nucleotide bases. So this is very easy. Nucleotide bases are A, T, G and C. They are also put into the same tube that in which the polymerases are put, and nucleotide bases will be put, primers will be put, and again buffers would also be put inside that. So we have talked about nucleotide bases. Again, nucleotide bases would be A, T, G and C. Nucleotide bases would be A, T, G and C, right? So now coming to primer. So this is again a very important aspect of polymerase chain reaction. We have to deal it in detail. So let us talk about primer. So what are primers? They are short stretches of DNA that bind to your template at a particular position, and then from there they provide the free 3' OH terminal from where the DNA polymerase starts, and it tries to elongate or it tries to extend this primase, right? Primer, right? So in this case, primers are designed from beforehand only, and a number of in silico methods, a number of computational softwares are now available for designing particular primers for your particular reaction, for your particular template that you need to amplify. Okay. So one very important thing, one very important disadvantage for polymerase chain reaction is that you need to have a prior information about your sequence. Then only you will be able to design your particular primers. Suppose I will show you this in bit. Suppose this is the DNA strand that you wish to amplify. You have to get the information. Suppose this is from 3' to 5' end, and another strand is present which is from 5' to 3' end, right? So you have to have the information about this particular stretch of DNA, because you will have to design the primer for this. Similarly, for this region also you need to get the information about this region, because you will have to design the primers. Primers would be complementary to this particular region and this particular region. They will go and bind to this template and would provide a 3' OH free 3' OH end, and this would be used to extend the this template. Okay, the extension would be performed by using this primer. Okay. So there are multiple softwares, computational tools that are used for construction of primers, but you need to remember a few things, that the primers, before choosing the primers, you should be aware of some important things I would tell you here, that primers should not be self-complementary. Why I am saying that? Because you will be using a forward primer. Suppose this is a forward primer, and this one would be a reverse primer. You would be requiring two primers, and these two primers, that is the forward primer and the reverse primer, should not be self-complementary, because all these things are added inside one tube only. If both the forward and the reverse primer are self-complementary, what will happen? They won't bind to the template, and they will just bind to themselves only. Okay, so this would be clear. Another important thing is, no hairpin structure should be formed. Okay, so the primer should not have a structure in which they try and fold on themselves, just like this. they should not form a hairpin structure, or they should not bind to themselves only. So this should again be an important factor that should be remembered while designing the primers. Next important thing is GC region should be kept to a minimum at 3' end. 3' end GC region should be kept at a minimum. Okay, and temperature for annealing, temperature of annealing the primer should be far away from the denaturation temperature. These are the 3 steps that we would deal in detail when we come to the steps of PCR. So the step of a particular, the temperature of a particular step, that is denaturation, should be far away from the temperature of the annealing of primer. Suppose the temperature for denaturation is 94 degrees centigrade, and suppose the annealing temperature is 72 degrees centigrade, then it is okay. But say the annealing temperature is near to the denaturation temperature, then it would also create a primer. So these it will also create a problem. So these are some of the important points that you need to keep in mind before designing the primers for your reaction. Okay. So with this we complete the primer section. Now coming to buffers. So it is important for any reaction, it is important to keep the reactants that are taking place, that are involved, in a suitable environment. That is why buffers are used, and some of the cofactors are also used because DNA polymerase is utilizing those cofactors for the elongation of our template. Okay, so let us understand this. So this is a PCR tube. Let us understand this. This is a PCR tube. What all things are present inside this? First of all, the most important thing is polymerase. Next one is nucleotide bases. Next is primer, and then template, and then buffers, and then cofactors. Right? So template, again, I haven't told you about template. I haven't written template over here. So template is the strand. Template is the DNA that you want to amplify. So template is also present inside. Each of these factors, each of these molecules are put inside this tube, these PCR tubes, and this PCR tube is then put inside the PCR machine. After putting this into the PCR machine, we would now understand what are the steps of PCR. Inside PCR machine, what happens, we will understand this in this region, that is steps of PCR. Now let us understand steps of PCR. Okay. So the first step that we need to understand is let us understand is this in a diagrammatic way, right? Steps of PCR. So suppose this is the template. This is the template that we want to amplify. We already know that the DNA strands are anti-parallel. Suppose this is in the 3' to 5' direction, and this one is the 5' to 3' direction. Okay. So this is the template strand that we are working upon. In the first step, what do you do is denature this double helix, right? So how do you denature the double helix? You denature the double helix by heating it. Denaturation. Denaturation would be utilization of heat, utilization of temperature for breaking these strands, for breaking the hydrogen bond that is present between the strands. When the hydrogen bond will be broken, these two strands would move apart, right? So what would be the temperature of denaturation? It would range from 92 to 96 degrees centigrade. So this temperature and time are basically little bit variable according to the program that you are using, according to the template that you are using. It also depends upon that. Suppose you have a high GC content in the in your template, then you would require a high temperature for the denaturation purposes and so on. So this is variable. We would just take the average, or we would just take the most common ranges that are used for a PCR reaction. Okay, so in first step, denaturation occurs. What happens in denaturation? These two strands that previously were stacked together, that previously were hydrogen bonded together, they will separate. So now these two strands would separate. This is again 3' to 5'. This is again 5' to 3', right? So denaturation has occurred. Now what will happen? You have to go for the next step. You would have to go for primer annealing. You want to now attach your primer to these regions so that the further processes, so that the elongation should be continued. Okay, so now we would go for primer annealing, right? So the primer annealing also would require a particular temperature that would be of 55 degrees centigrade, right? So primer annealing would require a temperature of 55 degrees centigrade. Now how will the primer annealing step look like? Suppose these are the two strands. These are the two strands. Now let me basically I would draw from here. I would continue over here. What do we mean by primer annealing? These are the two strands, right? Now we have to put the primer over here. This strand is from 3' to 5' and 5' to 3' and we need to put our primer in place. Suppose we have attached our primer here and we have attached our primer here. So this is what we mean by primer annealing. So primer would be attached to these particular regions, right, at particular temperature. So this was what we mean by primer annealing. Now again we want now to elongate these the extension of, we want the extension of this primer in this direction and in this direction. So you know that DNA polymerase elongates or has a polymerase activity in 5' to 3' direction. So how will this take place? So this would be the 5' end and this would be the 3' end, and here this would be the direction of extension. Again this would be the 5' end and this would be the 3' end of the new strand that is formed, and this the black template has the same, the same the same ends, that is 3', that I have just copied the template strand over here and just I have shown the direction of elongation in the new strands, right? So this is how the elongation would take place, and after completion the two strands would be differentiated, or the two strands would be separated out. Okay, so this is the process of elongation. Elongation takes place at 72 degrees centigrade, right? So we have three steps. And again after this process, what do? We again go to the denaturation. So this is just like a cycle. After elongation, then again denaturation will take place. These two strands will again separate out and we will get amplified DNA. This is the cycle that would go on continuously, right? So this is these are the steps of PCR. Now let us check the amplification. Now before checking the amplification, one more thing that I would like to show you, that how can we say that amplification is taking place. Okay, suppose we have these were the two strands that we started with, right? What are we now expecting? We have undergone, what have we undergone? We have done the polymerase chain reaction. So as I had shown you earlier, that one of this particular strand would again serve as a template for the another strand, and this would also serve as a template for another strand. Let us understand this. So this is the old, red one is the old or the original strand, and now with the help of this red one, we will again get a new strand. This new strand has been shown in blue color. This has been formed by PCR, polymerase chain reaction. Similarly in here also. Now these two strands will again serve the template for another polymerase chain reaction cycle. Just like this. Here just see carefully that here the original template strand was used to form new strand, and this strand was again used to form the new strand. Therefore here both are blue, right? Okay, so this was cycle 1. Okay, this is cycle 2. Okay, so after each cycle, what do you see? After this also, these will form again 2, these will form again 2, these will form again 2, these will form again 2. So what can you conclude from this? That after each cycle, you can calculate the number of the DNA double helix that are formed from each DNA molecule. And how can you calculate that? With the formula of 2 to the power n, where n is the number of cycles. Here we had started from 1, and after 2 cycles, how many DNA strands how many molecules of DNA that we have obtained here is 1, 2, 3 and 4. How can you calculate this? You can just put the number of cycles value to be 2, and you will get is 2 to the power 2 would be 4, right? So this is the basic from here numerical questions also come from CSIR, for the CSIR examinations, right? So this is what I wanted to show you about the process, that how amplification takes place. It grows exponentially, right? It carries on exponentially. So that is why we have used the formula 2 to the power n, right? So this was about the steps of PCR. And one more thing I would like to tell you, that you might have a question in mind, that how long, for how many cycles this process can continue. So this basically continues, a particular PCR reaction can continue for generally or about 30 to 40 cycles. And why does it stop after 30 to 40 cycles? Because the DNA polymerase activity starts going down, because the polymerase tube, the Eppendorf tube or the PCR tube that you are working in has a lot of now, because of the DNA amplification, it has a lot of DNA. The tube becomes a lot more populated, a lot more DNA content is present inside the tube. This basically reduces the activity of the polymerase enzyme that is present, and due to the reduced activity of polymerase, it generally ends around 30 to 40 cycles. Okay, you can enhance that by using different polymerases, or you can enhance that by modifying your program to some extent, right? So we have talked about the steps of PCR. Now let us talk about checking amplification. How can you check that your DNA that you were working upon, that you wanted to amplify, has actually been amplified or not? You can do that by running a gel. How can you do that? Suppose you have a gel. This is a DNA gel. Remember that this is a DNA gel. Now in the leftmost corner you run your primer, right? Suppose you start from 200, 300, 400, 500, 600 and 700, right? suppose I've already told you that you need to have a prior information about the sequence that you are amplifying. Suppose the target sequence had a DNA molecular weight of around 500 kb. So after the PCR, after the tube, here you had got all your mixture. In this tube you had all your mixture, and you have taken this tube out from the PCR machine after the program has been completed. So you think that in this particular machine you would have your amplified DNA. Now you just have to check it. What do? You just take this amplified DNA, process it, and then you just put it inside the wells of DNA gel. Now, if you were thinking that the sequence that you wanted to amplify was around 500 kb, this here you would get a thick band of 500 kb, a proper band of at around 500 kb. But suppose you did not perform the experiment as per protocol, or something went wrong in the experiment, what would you get? You would get a smear just like this. You won't get a solid band at the required or at the relevant site. Okay, so this is how you can check your amplification with the help of a gel. Okay, so this was about checking amplification. Now let us move on to advantages. There are a lot of advantages of PCR machine. It is a very useful machine and is used routinely in molecular biology laboratories. In current scenario of COVID, RT-PCR machines are used heavily, and they are used for determining that if a particular person is infected from COVID or not. So advantages, first is it is highly robust. It is highly sensitive. It can work with small quantities of DNA also. And it can also work if the DNA is not proper or has been degraded to some extent. It can also work on that. It is rapid. Okay, and it is complete, and it is comparatively not as much expensive as a cloning technique. Okay, so it is not as much expensive as a cloning technique. So these were some of the advantages that that a PCR machine has. Now let us focus on disadvantages. When we come to disadvantages, there are not many disadvantages that I can think of. First and the foremost disadvantage that I can think of is that you need to have a prior information about the sequence. You need to have a prior information about the sequence, then only you can design the primer, then only you can go on with further processing. Okay. So another important disadvantage that we can consider is the process is long. Suppose it is for 5 or 6 hours process, and when you can just check your product after you run the gel. You cannot check in between. Whatever happens in your reaction happens in your reaction mix. You cannot check it in middle, and you can just check it after your DNA is run on the gel. So this is also an important disadvantage. Furthermore, some contamination can also be seen in case of PCR machines, in case of PCR reactions, and you can get false positive results also. Suppose for long templates, if your primer has not been designed properly, your primer can bound to non-specific places, and then you can get non-specific results also. So this is what is contamination, and this was all about the PCR. This was complete lecture on PCR. If you liked my lecture, please hit the like button and subscribe to my channel. If you have any queries related to this chapter or my previous videos, please comment on the comment section, and I would aim at sorting it out in the next upcoming lectures. Thank you all. Thank you so much.